RNA transcription. So this is how we use the DNA in order to make RNA. So similar to DNA transcription, helicase unwinds the double-stranded DNA, and then topoisomerase prevents supercoils. But different to different to DNA transcription, RNA transcription doesn't need a primer. So in DNA transcription, you need an RNA primer. But since you're making RNA, you don't need a primer. RNA polymerase can bind to the promoter regions in DNA. This is aided by transcription factors. So just like DNA replication, the template or antisense strand is used to make the single-stranded mRNA. So it's good to note that the template strand is always antisense, and the mRNA that you make is identical to the coding or sense strand. So if you see a template or antisense strand, the mRNA created will be the opposite base pair to that. So if the template has like an A, the mRNA would be a U, while if it was a G, then the mRNA would be a C. And then, so after the RNA is transcribed from the DNA, it's called heterogeneous nuclear RNA. So that's the primary transcript that's formed, but before it can leave the nucleus, in order to be used in protein synthesis, it has to go undergo a bunch of post-transcriptional modifications before it's ready. So that's when they edit the mRNA. So they'll splice it, they'll cut out unneeded regions in order to create a specific mRNA that creates a specific protein. And so another type of RNA is ribosomal RNA, or otherwise known as RNA. So rRNA is also made in the nucleus, but it's made in the nucleolus. And so that this is an RNA that's being used in ribosomes. So RNA, it's used to make ribozymes, so which is an enzyme that uses RNA instead of peptides. So it's an enzyme that instead of amino acids, it's made of RNA. And this RNA, it's used in ribosomes, and it's used to catalyze peptide bond formations. So it's actually connecting separate amino acids through peptide bonds to form the full protein. So ribosomes, they're made up of rRNAs and proteins, and they have 3 binding sites for the tRNA that holds the amino acid to make the protein. So the first site, it's called aminoacyl. Then other site is P or peptidyl, and then the last site is E exit. So ribosome has 3 different sites for the tRNA. And I'm going to go over RNA translation. So how is the RNA used to form proteins? So RNA translation, so first thing, initiation, the ribosome binds to the mRNA sequence that was transcribed. And like I said before, it brings it together with the charged aminoacyl tRNA. So that's the tRNA that is bounded to a amino acid. And when it brings it together, so when it brings the mRNA and tRNA together, if the codon and the anticodon match up, it will add the amino acid to the growing chain of the polypeptide. So the first codon always has to be AUG. So AUG is a start codon. So the chain where the mRNA sequence where there's an AUG sequence is the start codon. So that's where the ribosome will start translating the mRNA into a polypeptide sequence. So RNA translation. So the next step is elongation. It's a 3-step cycle that's repeated for each amino acid added. So the ribosome, it moves in the 5' to 3' direction along the mRNA. And the protein is made from amino, or the N terminus to the carboxyl or C terminus. So the peptide bond that's formed, it's formed from amino to a carboxyl terminus. And that's why it's called a peptide bond, because of the amino carboxyl bond. So the peptide bond is formed by peptidyl transferase. So peptidyl transferase is the enzyme that forms the peptide bond between the 2 separate amino acids to form the polypeptide chain. So like I said before, there are 3 sites in a ribosome for the tRNA. So the A site, it holds the incoming amino acyl tRNA complex. So it holds the next amino acid that we added, and that's determined by the mRNA codon in the A site. So if the tRNA has a matching anticodon to the mRNA codon in the A site, then the peptide or the amino acid will be added to the polypeptide chain. And then the P site holds the tRNA connected to the polypeptide chain. So each time a amino acid is added onto the polypeptide, that tRNA will move into the P site, and that tRNA holds the growing peptide chain. And once the amino acid from the tRNA is added onto the polypeptide, the tRNA is moved from the the polypeptide is passed from the tRNA in the P site to the tRNA in the A site. So like I said before, each time an amino acid is added to the polypeptide, the tRNA will transfer the polypeptide to the next tRNA in the A site, and then that tRNA will be moved into the P site. And then the last site, the E site, that holds the inactivated or uncharged tRNA. So after the tRNA gives up the amino acid bonded to it, to the growing peptide chain, it'll be moved into the E site before exiting the ribosome. And then that tRNA that left, it can be reused again. So it just has to find the matching amino acid, the amino acid that matches the anticodon in the tRNA, but it has to be a specific amino acid, since each anticodon is only specified by one amino acid. And then last step, termination. So once a stop codon moves into the A site, so once the stop codon moves into A site, the release factor RF protein binds to the stop codon. So the stop codon, they don't specify any amino acid. So these stop codons don't have a amino acid correlated with them. So the mRNA, the stop codon, it will bind to a matching anticodon on the tRNA, but then the tRNA does not hold any amino acid. What happens is the release factor adds a water molecule to the polypeptide, and that's used to hydrolyze the tRNA from the polypeptide. So that releases the polypeptide from the tRNA in the P site, and then the polypeptide is complete after its release. And since the protein is formed, the ribosome subunits dissociate until they encounter another mRNA that's ready for protein synthesis. But before the polypeptide chain can properly function, it has to undergo post-translational processing. So things like chaperone proteins has to bind to it in order to fold it into the proper shape. So proteins, they need a specific shape in order to function. And chaperone proteins help that by allowing it to fold into its proper shape. And then some proteins also go cleavage events. So the polypeptide chain has to be cut down before it can be active. So a large inactive protein will be cleaved into a smaller protein, but that activates that protein. So the polypeptide chain will have to be cleaved by an enzyme in order to be activated.